Hello. Today I'm going to be talking to you about how to use Sigma plot and how we use it in our research lab to determine the IC50 value for our compounds. So the first thing is that this video is going to be in three parts, just to keep things short and easier to swallow. The first part is going to be all about sort of the background behind this data and where it comes from and how to organize that data to use it for Sigma plot, right? Then the second part of the video will be about actually using the Sigma plot software and how you use that to make the graphs and the curves and find your IC50 value. And then finally, the third part of the video will be about formatting and how to properly format your graph and titles and axes and all that good stuff. And also a little bit extra stuff about how you can make your graphs look really professional and combine them. So let's go ahead with the first video here with the background. So Sigma plot is this very powerful software that allows you to graph and do data analysis on lots of different types of data, right? So we're going to use it in conjunction with Excel, but pretty much any software that comes in the form of a Excel sheet, right? So Google Sheets, anything like that, you can transfer that data into Sigma plot. You'll see that in the second video. What's really great about Sigma plot is that it does a lot of the math for us. So as long as we get the parameters right and we tell it exactly what to do, it does all those transformations and analysis for us. What we are specifically interested in, down here at the bottom, oops, let's go back, you see this IC50. That is defined as the concentration of our inhibitor that is required to inhibit half of an enzyme's activity. So if you look at this graph right here, all the way in the bottom right, you can see these three sort of curves that are generated from these three sets of data. And this is sort of what you'll be generating in Sigma plot. A little different, but close enough, right? And you can see this goes from 0 relative fluorescence all the way up to 1. So at 1, it is fluorescing at its full amount, right? And at 0, it is not fluorescing at all. As you can see, as you are increasing the concentration of your inhibitor along the x-axis, the relative fluorescence is decreasing, right? That makes sense. It's producing less of your fluorescent compound, whatever that may be. And so less fluorescence is happening. And so it's a little bit easier to sort of figure out what's going on by looking at these graphs. What we're interested in is the IC50, right? So the 50% concentration mark. To find that, the software does this for us, but it basically looks at the point where 50% of your fluorescence has been sort of removed, right? So this is at exactly 0.5 relative fluorescence. And then it draws sort of the straight line over to your graph and then straight down, and it tells you whatever concentration that should be, and that is your IC50 value, right? That concentration is IC50, okay? So where does this data come from, right? So we do what we call a bioassay, which is it's aromatase inhibition test. And you can see this long sort of process that it goes through, right? What we are really interested in is the fact that we, down here, you can see we have all of our test materials, and if you get trained on the bioassay, you'll understand what all this means. But effectively, we put a fluorogenic substrate, right, into our test kit, along with our inhibitor in concentrations as you increase along the plate horizontally. And what happens is, as this goes through this process, it creates this fluorescent product, 7-hydroxy trifluoromethylcoumarin, right? It goes from 7-methoxy trifluoromethylcoumarin to 7-hydroxy, right? So it's hydrolyzed. And what that does is it changes this substrate from something that doesn't fluoresce into something that does. And so that allows us to sort of tell how well this inhibitor works, because if more of the fluorescent compound is created, it is not inhibiting this enzyme that well. And this enzyme that we're looking at is human aromatase, and that's CYP19A. That's the code for human aromatase, right? What's really important about this is that you understand that we are going from something that is not fluorescent into something that is fluorescent. And so we are measuring the amount of fluorescent product that is generated. This down here in the bottom corner, you can see this fluorescent plate. This is what we do all of our experiments on. And so when you see me sort of talking about the wells and the data in a couple slides, you will understand that that is directly related to each well on this plate. Each cell in Excel that you are going to see corresponds to one of these little wells in this plate, right? And so you have 8 columns and 8 rows and 12 columns. And that's very important because that means we could potentially do 8 compounds because we put each compound along one whole row. So we could potentially do 8 compounds per plate, right? So after we do all that bioassay stuff that you don't really have to worry about, we take it to our fluorescent plate reader, and it uses this SpectraMax i3 software. And this is sort of what it looks like after it's finished running. And what happens is we save this data in 2 forms. First, we save this file down here called SDA. That is the file that the spectrometer can read, right? The fluorescent plate reader can actually read. So you always want to make sure that file is saved. You don't want to delete that. That's the file if we ever needed to open it back up in the software for any reason to look at things, we could do that there. We also save it as a text document. And as you'll see, that is the document we actually use to put our data into Excel and to start manipulating it into a form that we can then take into SigmaPlot, right? So this is what it'll look like at the end, but we're not going to look at this yet. We're going to go straight to Excel, and we're going to open up a blank sheet here. Put this at the beginning here. The sheet should be blank, so let's delete all this. There we go. Okay, so we have this blank sheet, and what we're going to do is we are going to insert our data, right? So to do that, what we do is we come up to the Data tab in Excel, and you can either click this Get Data from File, right? And then come over here from Text/CSV, or you can simply click the From Text/CSV button if you can see it. Click that. And what it'll do is it's going to open up a file browser, right? Normally it'll be at the very first file. So I'm going to start all the way from here and click through, and we're going to go through into my folder, the video settings, into the biochemistry here, into here, and then the very first one. And you see there are these 2 text documents, right? So this was 2 runs of the same plate, and so we're just going to open the first one. So you select whatever text document you want, and I just clicked through all that so you could see where this one is if you wanted to follow along. But you can open any text document, really, as long as it has the data you want in it. So you come down here, and once this is selected, you click Import. Wait for it to load for a second. And now what you're going to do is, this is not how we want it to look, right? Because it's mushing all this data together. So what you do is you click down here on Transform Data. Okay, and it's going to sort of show you what it'll look like here, right? So if we just closed this and loaded it, it would just kind of put it all in one big block, and that's not what we want. So we come over to Transform, and we want to split column by delimiter. So delimiter is just something that it uses to tell you where the stops are. So we are using tab, but if you, for some reason, were wanting to use this for some other data that you were using or importing, you could change it to any of these. Some programs do it by spaces, some do it by semicolons or whatever, right? You can also do a custom. So if you wanted to input any sort of key character that you wanted to split all your stuff by, you could. We're doing tab, And we want to say that at every time there is a tab in, like the tab key has been pressed by the program, in this data, we want to split that into a new column. So you just click OK, and it'll do it for us, right? And you can kind of see what it's going to look like here. Now, you could click File, Close, and Load. The problem with that is it does a lot of formatting where it puts it in a table and it colors it and all that stuff. We don't really need all of that because really what we're looking for is just the raw data. What I like to do is I just like to click on the first cell and do Control A, which selects everything, and then you click Control C to copy it, and then you can close this window, just click Discard, and then in the sheet you want to paste it all, you click Control V to paste. And so you see it has a bunch of these null values because it's been splitting some of these pieces, but this is all of the data that comes off of that text file. So it's all in here. So the first thing I'm going to do is go through and start deleting all of this extraneous information, all these nulls, all these titles that we don't need, so to clean up this data a little bit, right? So going through and just deleting all of these null cells. After you have deleted all of these null cells, this is roughly what you should be left with, right? So it has the labels for what columns are which, right? So this is all column 1 on that plate, and this is all set out in one row, right? So what we need to do now is we need to label it by row. So normally this would, you would have like a notebook or someone's data of how they ran this. We're just going to go ahead and I'll have that's automatically put in for us here. Let's just delete this extra sheet here. So what you can see is this data came from the notebook of the people that ran this particular experiment. And so effectively, on the plate, each row is given a letter, right? And these letters are actually on the plate. It's a little hard to see in the picture, but they are there. So in row A, we did a 20 millimolar letrozole, and across it, we did all of this data, right? And so we have all this data in place, and this is now what we're kind of ready to process. So now it's labeled, so we're not going to lose track of like which row this was. But real quick, let's talk about what this data means. So back to the PowerPoint here, and I'll make this screen. So this is the same data that I just had on that other page, but this is a little bit easier to see. So on our plate, we usually do something called a positive control. So a positive control is some compound that we know 100% works, right? We know, and letrozole on this plate is our positive control. We know that letrozole should work, right? It should inhibit CYP19A, the human aromatase, and so there shouldn't be any fluorescence, right? Or if there is, it should be low. So that's our positive control, and this was sort of our first run here. So we had a 20 millimole and a 0.2 millimole run, and we did doubles of each. We also have a negative control. So this is where on the plate, so we serial dilute, so we put all of our compound in the first one, and then we dilute across each one of these. For cells 9 and 10 here, we don't put any inhibitor in those. So what that is, the point of that is sort of to say, okay, without any inhibitor, the aromatase kit is working and it is producing something that is fluorescent, right? And so if these cells were really low, then we know that we'd have to throw this data out, because that means that even on its own, it wasn't producing. Something is wrong with the process or the kit or the materials, right? So that's the point of the negative control here in the blue. And then finally, in the background, these are our blanks, right? If you've ever done a blank for an instrument, effectively there is nothing in this except for the solvents that we run this in. We use acetonitrile as our solvent. So in these blanks, it's just all the background materials. And what this is meant to do is the instrument subtracts these values for us from the rest of the data, an average of these two, because what these are saying is that even though there is nothing but acetonitrile and whatever dust or whatever's in the air in those cells, it's still getting a tiny bit of fluorescent reading. Maybe that's reflection off of another cell, or some random light artifacts that are around, right? So these blanks help kind of tell the instrument where our zero value should be, and so it helps make everything else pretty controlled. So don't worry too much about the blank, because the instrument actually takes care of that for us, the background, and it deletes that out from everything. Some instruments might not do that, so if we change instruments in the future, usually the way you do this is you take an average of these two values and subtract that from all of these values. Again, we don't have to do that because the instrument does it for us, right? So let's go back to the Excel file. So we just saw that that's sort of how we want to do it. Next, however, is this is where we're actually going to manipulate the data slightly to get it ready for SigmaPlot. So the first thing I like to do is I like to copy and paste whatever the row I'm going to be working in, right? So I'm going to do this first row real quick. So the 20 millimole of letrozole. Now, in SigmaPlot, which you'll see later, it really prefers to work with data in vertical columns. It doesn't like that our data here is horizontal. So what we need to do is we need to transpose this data into a vertical format. So what we do is we select all the data we want to transpose. So we're going to do cells 1 through 8. And there's a couple ways to do this, but the way that I find is easiest is you Control-C to copy it, and then instead of hitting Control-V, which would just paste it as normal, instead you select your first cell, you right-click, and then you do paste right here as transpose. You can also do paste special, and transpose will also be in there as well, right? So just click transpose, and then we'll make sure that this data is all nice and centered. There we go. And now our data is in a way that SigmaPlot kind of likes for us to be. Now there is one more piece of this that is a little important, and that is rows 9 and 10. So remember that 11 and 12 we keep because it's important, but we don't need this. The instrument just reports blank values. We do want the values for 9 and 10, and we want an average of those because that's our negative control. So if you think about it, that is technically our zero concentration of inhibitor. And so we end up putting that in the data as sort of a comparison point, right? So to do that, we want to get an average of these two values and put it at the end of this, right? So if you have, this is 1, 2, 3, 4, 5, 6, 7, 8. The last one is actually going to be 9 and 10, right? The last value here will actually be the average of 9 and 10. So what we're going to do is you can do this in any way you like. I just like to off to the side somewhere just to do equals AVERAG equals average, open parenthesis, select the two data you want to average, close parenthesis, enter. And now this gives you an average of these two values. And if you do it exactly like this, actually what's really nice is you can just drag this down and take the average of all of those values, right? And of course, it's always good practice to label if you do this for anything. 9 and 10, right? And so it's always good practice to label your data so you always know what you're doing. But now what we can do is Control-C to copy, and then when you paste it, you want to make sure you're pasting only the value. You don't want to paste the actual equation because then it's not going to be right? It's going to pick some other cells. So paste the value, and there you go. And then you just repeat that over and over for all of them. So I'm going to do that real quick. Okay, so I've gone through and I've done this for all the other rows, so we're ready. There's one more thing that I did that you can do as well, and I highly recommend you do, is I went ahead and I put in the concentration that each one of these cells should be at. So this is actually a very good thing to know. We generally do this whole experiment at 20 millimole concentration of whatever all of our things are, right? So the concentration in millimole of this first well is 0.4, right? And then the way you get the next wells after that is all you have to do is divide this first one by 3. So if we have 0.4, right, and then we say that the cell above divided by 3, we get the next one, right? And you just drag that all the way down, and all these values are the same, right? Because we are just doing a 1/3 dilution each time. So you don't have to worry too much about calculating this by hand. As long as you remember that 0.4 is the first well, and then you divide it by 3 for each subsequent well. For 20 millimolar as you're starting, that's great. Every now and then, as you can see up here, we may have something that is actually 0.2 millimole or even 2 millimole or some version of that. To fix that, all you do is you just move the decimal here as many as need be. So if, for instance, we were working with this 0.2 millimole letrozole compound, that is 2 decimals away from this, right? So what that means is our starting concentration would be 0.004, right? Because it's 1/100th of the concentration here, so this would also be 1/100th of the concentration here, right? So, yeah, 1/100th. And then again, you would just do the same thing where you divide this by 3, and that's all you have to do to get this concentration value. So it is always a good idea to know exactly what the concentration of your inhibitor is, so that when you do this part of the data, you make sure you're putting the right concentration value next to it, because this will also affect your final IC50 value. But this is it for processing the data. All of this is now ready to go straight into SigmaPlot. So thank you, and I will see you in part 2.